started out with the premise of closed ecological system. And as I started working on the diagrams, I began to realize that if we're going to do this ecologically, we had to have selected species for each of the niches. But I also realized as I started adding creatures that each creature had to add a certain number of contributions to the overall system. Black soldier fly, for example, they consume human waste. They're a pathogen consumption point and blocker. And also they provide food that goes down the chain to the worms. The worms provide not only food for the quail and the fish, but they then create the soil that we will then use to expand the garden. And then, of course, the fish provide food for the plants. They also provide food for us during harvest. The spirulina will clean our air, but also then provides part of the feedstock recipe for the fish and the quail. The idea for the project was the assumption that if I'm living on Mars and a supply ship fails to reach us, we must be able to produce sufficient nutrients as well as longevity within the ecological system without dependence upon the supply ship. Microbiological stuff is great, and we're going to need that, but we've got to go beyond just the microbiological. Next year, we're actually hoping to increase the last piece, which is mushrooms and mycelial colonies, in order to enhance the garden and the growth processes and so forth. Yeah, some of it was from The Martian. The last slide, if you go to our website at www.earthseed.space, there are links to our Facebook, email, and LinkedIn on the front page, as well as a lot more information about what we're doing.
Great. And is that the best place to contact you to learn more about the MASH?
Absolutely. It's a little outdated as we just repainted the MASH. The paint job you saw in the pictures is brand new, barely 2 weeks old.
That's cool.
Some of you may remember it was a big black behemoth before, but we've added this so that we can travel. We are also, just a side note, I'm having discussions with the world's biggest analogs in the hopes of becoming an outreach function and visiting the 4 habitats here in the continental US as part of that outreach process. And so I'll be driving, and I will be in sim, which means every time I get out to get gas, I'm going to be in a sim suit. So we're hoping for lots of questions and answers from the general public. Thank you, Patrick, for the website link up there.
Yeah, of course. Thanks for the excellent presentation, Ducky. It's you're in the right place. All of these people are here for just this purpose, talking about these closed ecological life support systems. So we're really glad you could share with us what you're working on. All right.
And just
go ahead.
As a last note, I was just going to say, if you look, I just turned my video on for just a moment. The cabinets behind me are actually above my bed. I am in the MASH at the Mars Desert Research Station using their Wi-Fi to transmit this presentation. So you got it straight from the beast right here today.
Yeah, that's the exact application. That's really cool. Thanks so much for making the time.
Thanks for letting me in.
Yeah, of course. Okay. We are not really on schedule anymore, but I think it's fine to just proceed. Yep, you're up next, Morgan, with endophytes for increasing plant resilience and survival under abiotic stress.
Awesome. Now have the obligatory Zoom question of, can you see my slides?
Yep.
Okay, great. Okay, cool. Good afternoon, everyone. I'm really excited to be here. It's my first Space Ecology Workshop, both as an attendant and a presenter. And I'm happy to share my research on endophytes for increasing plant resilience and survival under abiotic stress. So my name is Morgan again. I just began my third year as a PhD student at the University of Washington in both the Environmental and Forest Sciences and Astrobiology department. So I spend my time researching and thinking about plant microbe interactions at the individual scale, but also how the plant microbiome shifts under abiotic stress at the community level. And as we explore the possibilities and potential related to space ecology, it is important to discuss some challenges that we are currently facing on our own planet. So the IPCC stated that current changes in global climate are starting to result in irreversible trends. 2023 was the hottest year on record, and we are currently on track to surpass that in 2024. So this transition to sustained heat poses particularly significant challenges for plant survival and ecosystem stability. Heat stress affects organisms in a variety of ways and has both intermediate and long-term consequences. As we consider the future of plant cultivation, both here on Earth and elsewhere, it is important to understand the responses to climate change, as we are essentially witnessing the extremification of environments. So, what happens when we inevitably heat up? Now, I'm not a human physiologist, but we can think about the symptoms of heat stress using a human metaphor. So if I presented you with these two individual symptoms, would you be able to tell who is experiencing excess heat and who is experiencing generalized stress? And the reality is it would be difficult to tell because the symptoms related to excess heat and the symptoms related to stress are frequently overlapping. And it's the same story with plants. So where environmental stress induces non-specific responses, such as photosynthesis being disrupted via damage to thylakoid membranes and chloroplasts, which would reduce carbon fixation efficiency. Plants often activate antioxidant systems to combat the increase in reactive oxygen species that can affect cellular processes. And an increase in transpiration can lead to water loss, which would then induce the closure of stomata, leading to a decrease in carbon dioxide uptake. And overall, these responses would slow or entirely stop plant growth. So we can use heat stress as a proxy phenomenon for studying the mechanisms related to coping and survival under stressful conditions like microgravity and radiation that would be introduced during spaceflight, which is good considering the difficulty in accessibility of actually testing plant growth in microgravity conditions. So we can use this information to elucidate ways to alleviate the plant becoming overwhelmed by these responses. Ultimately, excess stress causes damage to plant tissues that can result in plant mortality at both the individual scale, but also at the population level, such as crop failure and forest dieback. Another consequence of this is leaving the affected plants more susceptible to pathogens or other stressors as well. So understanding and mitigating plant stress is an important factor in the success of long-term space exploration and potential colonization efforts. And relying on plants that are already adapted to heat or environmental stress would be, I think, a difficult solution here. So space environments present multiple unique stressors and resource limitations. And while some traditional approaches like genetic engineering or selective breeding of plants has shown some success, it can definitely be a lengthy and resource-intensive process that can also lead to some trade-offs between growth and stress tolerance, where often plants bred for stress tolerance have greater resilience, but then reduced productivity, which is important to consider depending on the motivation and the types of plants that you would choose to grow in spaceflight. So we can use the relationships that plants already have with endophytes, the beneficial microbes that reside in the internal plant tissues, to solve some of these challenges. Endophytes have been documented in a variety of contexts to aid in plant health, and they possess some advantages over relying on soil microbes alone. So especially in a space ecology context, they have direct interactions with plant tissues, and they have co-evolved with their hosts and evolve faster due to bacterial generation times when compared to relying on plant adaptations. Endophytes isolated from natural systems can be introduced into agricultural varieties due to their broad host range. There's also evidence that inoculating plants with endophytes doesn't have to happen repeatedly, as they can be retained between plant generations. And because they have these internal relationships with their hosts, they aren't as directly reliant on the substrate composition, especially when you consider endophytes residing in leaf tissue versus root endophytes or soil microbes. So these endophytes can be used to bolster plants against the abiotic stress that they experience during spaceflight. And it has future applications for growing plants in lunar or Martian regolith. By strategically introducing beneficial endophytes, we can enhance and optimize the plant microbiome for surviving in these extreme environments. So I'm going to take you through an experiment briefly that we conducted to, one, build a collection of potential stress tolerance conferring endophytes, and two, a method to actually screen for that potential. So to do this, we collected above ground tissue samples from a black cottonwood tree growing on the hot, dry region of the Yakima River, east of the Cascade Mountain Range here in Washington State. This species of poplar typically grows along these riparian zones, but usually on the cool, west side of the Cascades. So we argue that because this tree is growing outside of its typical ecological niche, it may possess endophytes that help it to establish and persist in this region. So we then isolated several bacterial strains from these poplar tissues and extracted their DNA in order to further characterize them. At this point, we were still left with about 14 candidates. So we conducted a novel screening study to identify which of these bacteria are capable of bolstering these plants against heat stress. Each plant that we used in this experiment was an internally sterile poplar, so that its own microbiome would potentially not influence the outcome of the individual endophyte that we were testing. And each poplar was inoculated with a single endophyte and then placed in these constructed three-tiered vessels. And the vessels were connected to a tubing network that was pumping in continuously dry, hot air to induce stress. And we were reaching temperatures of about 38 degrees by like week 6 of an 8-week experiment. And with that, we were able to identify at least 2 endophyte strains that had a larger difference in plant mass when compared to controls, uninoculated controls. And this indicated that these plants were able to alleviate some of the stress and continue to grow despite persisting in these hot conditions. And I actually repeated this experiment and completed the breakdown process just under 2 weeks ago to follow up on some of the potential candidates for heat stress tolerance. And it appeared in this experiment, it appeared there is going to be like an even greater difference between survival and growth that can help us to elucidate some of these potential mechanisms. So maybe Space Ecology Workshop 2025. And when we look at the whole genomes of these Yakima River endophytes, we discovered that 11 of the strains were identified as potentially novel. And when we take a targeted exploration of their genomes, we see that some of these strains possess genes like ACC deaminase. So this is an enzyme that's an ethylene regulator and has been characterized for its ability to help plants cope with abiotic stress. And this mechanism is an advantage to the plants by decreasing the presence of excess ethylene that could damage tissues, but the bacteria also utilize this ACC as a nitrogen source. And ethylene reduction is just one example of the pathways these strains possess. There are actually several potential mechanisms to tolerating environmental stress, and the 2 strains in our experiment that perform the best are likely taking advantage of different pathways due to their genetic differences. So the generation of reactive oxygen species is a documented response in plants experiencing spaceflight, and there's evidence that this could be mediated by the presence of endophytes. We talked a little earlier today about increasing water use efficiency as an important factor to consider, and stomatal conductance and abscisic acid production can be modulated through endophyte activity. And it's not shown here, but additionally, in the context of microgravity, enzymes can produce osmoprotective molecules like proline to address the altered fluid dynamics that would affect nutrient and water distribution. And it appears there is an association between heat and UV tolerance, which could inform some choices for selecting endophytes in a high radiation environment. And we can use this information to inform the construction of a consortium of endophytes that would increase nutrient uptake and acquisition, increase water use efficiency, promote plant growth, suppress pathogens, and confer stress tolerance for the survival and success of plant growth in a variety of non-Earth conditions. Thank you.
Wow. Okay, Morgan, that was really, really cool. Where in Yakima were you sampling this?
It was near Ellensburg, in the
Hmm.
Yakima River Canyon.
Okay.
And this last year, I've been trying to monitor some of the weather and temperature conditions, anticipating it being an even hotter year than the year that these endophytes were initially isolated from, and it got up to be 115 Fahrenheit in July. So it's definitely a much higher temperature area, and lots of excess UV as well, just because it's isn't backed by the forest like some of the regular west side Cascade poplar that you see growing.
Yeah. Yeah. Wow. That is, I really like the thought process that you used to choose that tree in that location. It's really clever. While we're waiting for the questions to come in, I've got another one. You mentioned that for the controls you used internally sterile plants. How do you sterilize the inside of a plant?
Yeah, so this is a good question for Professor Sharon Doty. This is her wheelhouse of tissue culture. They're, I believe, initially grown from single cells and then
Mm,
continually propagated after that point.
I see. So not grown from seed, grown from tissue culture.
Yeah.
Okay. Yeah. Sharon chimed in the general chat. I guess that is
Okay,
correct.
good.
Wow, that's really interesting. Um, okay, let's see. Camilo asked which were the best genre that were isolated.
Yeah, so I think that's still, up for debate. There's definitely not a consensus that I've seen across the literature. But I'm mostly working with Microbacterium and Pseudomonas strains,
Mm,
couple Rhizobia, things like that. To be I feel like I'm being elusive here. I'm not, but I just finished this second round that I'm pretty positive about, that I think is gonna kind of help refine the answer to that
hmm
question.
hmm
I'm
Kind of,
non-committal right now.
yeah, that's understandable. In the same vein, Joya wants to know if you're thinking about doing combinations of microbes.
I would love to do that. Yeah. I'm actually in kind of 2 strains of thought. I'm interested in seeing what happens with this kind of synthetic microbiome if we did provide the plant with endophytes that um take advantage of a variety of nutrient sources for the plant, like nitrogen fixation, endophytes with the heat tolerance ones. But I'm also interested to see kind of the interactions or combinations that happen with a non-internally sterile plant. because I don't know how like feasible it is to continue to have a plant be sterile, especially if you're like cutting it repeatedly in a like, you know, space environment, not like sterile or not sterile, things like that. I'd be interested to see kind of like how the community structure in a plant changes with the introduction of like some of these endophytes too. So
Mhm.
yes, I would like to do combinations also.
Okay. I've got another question, or I guess maybe more of a suggestion.
Sure,
Um,
please.
my background here is biosphere 2. Uh, and while these days they don't do a lot of closed ecosystem experiments because it's not really closed anymore, when some of the research that they do is they look at the rainforest that's in there, which since it's in a big plastic, well, I guess a big glass dome in the desert, it's very, very hot up in the canopy, up to 50 degrees Celsius. And they use it as a model
Wow.
of what the Amazon rainforest might look like in, well, probably tomorrow or really soon, judging by the way things are going. Um, it could be really interesting to, I guess you're working with poplar, but if you found uh, a species that's in biosphere 2 and also in another rainforest, you could look at some other higher levels of temperature stress for your next endophyte.
Yes. Yeah, that's really interesting that you bring that up. I didn't mention it at all, but we do some work in the like tropics in like Costa Rica.
Hmm.
And it's a new program that we're like working with, but that is something I would be like really interested in developing too. Is, you know, I'm looking at these kind of like this hot, dry environment, but what is the hot, wet environment selecting for as well? Given that like humidity can kind of amplify some of the like temperature, like you said, up to 50 is very hot. So yeah, if you've got some connections that I can go take some clippings in Biosphere 2, I'll
Yeah,
happily
I
let you
mean,
know what I
I
find.
I definitely do. I did my master's there. The And usually those guys are at this conference, although I don't know that I saw any of the Biosphere 2 folks or SAM folks in the audience. Um, Christina's got a question, and it's about your funding, actually. I was kind of curious too. Which program is the Department of Energy funding this sort of research through? Are there other grant opportunities you know about in there?
So the one that I work under is it's using poplar as like a biofuel source. So
Mm,
it's like increasing the efficiency and like resiliency of poplar to grow on a like propagate is the wrong word, but on a like kind of agriculture or like plot scale. We've got some like pretty big trees. A lot of it that we've done previously is with nitrogen fixation. So decreasing reliance on, there you go. Thank you, Sharon. So sustainability to increase like access and efficiency of like using poplar for biofuels. So that's what this falls under. Yeah.
okay, that's cool. I didn't know we really considered trees for biofuel.
Yeah.
Yeah. All right. Well, I don't see any other questions. So I guess thanks a lot, Morgan. That was really, really interesting.
Thank
Um,
you.
yeah, we'll be excited to hear your updates next year.
Yes.
So we've got one more talk today. It was supposed to be Marina Lopez Pozo, but unfortunately, she emailed me yesterday, and she had like a pretty significant injury, which caused her to go for a unexpected emergency room visit. She's doing fine, but she's not here to present. So fortunately, Laura Fackrell had reached out to offer support in the case of some Hurricane Milton victims not being able to join. And we were really lucky that they were all here. But this is a good opportunity for Laura's presentation. So Laura, if you're around, I'm sorry, I don't remember the title of your talk. I guess I could find
Hey
it.
there. I think I just emailed it. Can you hear me all right?
Yeah, we can hear you just fine.
Yeah, so I emailed it a few hours ago for like the exact title, but I can pull up the slideshow because I think this will be the shift screen. So it's basically the talk is just an introduction to Rhodium Scientific, which is just a space biotech company, and trying to introduce some of the hardware that we have and some of the projects doing space agriculture in the space environment.
Well, take it away. We're excited to hear it.
Okay, perfect. So I'll go ahead and let me put this into actual slideshow mode. Can you still see the slides all right?
Yep.
Okay, perfect. All right, so this is a little bit different of a talk that I'm used to giving because I'm used to presenting lots and lots of data. I'm a scientist. My name is Laura Cockrell. I'm a staff scientist at Rhodium Scientific, and I just started here pretty recently in the last few weeks just to kind of get started on the more industry side of space ag. But I come from an academic side and the NASA side of space ag, doing lots of different work with during my dissertation, as well as during my postdoctoral NASA fellow postdoctoral scholarship, doing different projects that look at how we actually implement all these different things, the awesome science that we've been talking about today of growing plants in space and how you do that sustainably, and more of the different aspects of that, and how does the microbiome feed in, and how does that feed into an overall system and this closed ecological systems, and how do you make that all work together? And so I come from that kind of a science background, and so I'm used to talking about all the data from all the science I'm doing it. But today I'm going to talk about a little bit more of the hardware and a little bit more of that. So it's going to be interesting to kind of just be really excited about like what we're doing with all these different things. So to kind of give an overview of what I'm going to focus on, I'm just going to give a brief introduction to who Rhodium is, just in case you haven't worked with them before, or just to kind of give that context so you have an idea of where that's all coming from. And like kind of the commercial side of all of this kind of feature. So it's a little bit interesting. We've heard a lot of amazing science today. And so now we get to hear a little bit of how you implement that science through different commercial platforms. But I really want to focus on like what is the hardware that's available and like what are some of the products that work in it. So I'm going to get to that in a few slides, but that's going to be like the main focus. So Rhodium Scientific is a biotech company, and they do biotech both on Earth and in space, and are able to do that. And so just to kind of introduce who they are in general, is that they are a space biotech company. And so like there's a lot of biotech, and it's a really innovative industry in general on Earth, but now we have this new environment of space, and all these new challenges and things that are presented, and all the technology that's needed to kind of fill the gap, and to kind of and I think one of the reasons I think this is an important concept to introduce, and this is like it's useful to understand the commercial side of this as well, is that we have all these challenges that we've talked about today in the science that we're presenting, and we have all these different gaps that we're trying to fill in these science questions we want to answer. But one of the challenges is actually accessing the space and so, and how do you actually do that? And so, how do you take all these amazing questions that we're doing all this amazing work on? And we've seen a lot of talks that have done that and that have done this experiment in space, but it can be a big challenge to get your science into space. And so that's one of the things that Rhodium really tries to do, is provide that access to that space environment, and doing that through like high quality science and the hardware that we have that's available for them. So I'm going to talk a little bit about that, but they are just to give it a brief overview of who Rhodium is. They're a space biotech company that provides that high quality industry standard. And they've actually been able to, and there's a pretty strong team of scientists. And so they are very science based, not just in the engineering and like the technology side, but really the science that goes into that. And so it really has a huge focus on making sure that we're doing quality science in space, and that we're not just flying things to fly them, but that we are doing that in order to answer those really important questions and to really get them to help create that access to them. And there's been a lot of missions that have been able to do that. These are some of the ones that have been since 2020. And so there's a lot of different, we partner with industry and academia, we partner with DOD, we do, there's a lot of different things that we can do. And so we have lots of experience working with different kinds of partners and different, from academia to other small other companies to students, all sorts of different people, and getting access and providing that support and taking the science and translating it into that space and science is a really important part of that focus. And so really taking that industry standard and trying to understand and having those really quality like standard processes. And so not just putting something in space and slapping a label on it saying it was flown in space, but like really trying to make sure that we're doing the right protocols and having consistent and high quality assurances in the protocols that are given. So that's a really big focus of just trying to provide that process and trying to help scientists as maybe if you haven't flown, maybe you're working on a lot of the space ecology concepts, but you haven't flown something yet. We want to really help support your ability to make sure that you have a process and that the hardware you're using and the processes and the protocols you're using are really optimized for that purpose and getting to space. And so they actually call it the Rhodium Quick Space because they have developed a very particular command line of how you actually do this. And so that just kind of briefly presents like that's like that approach and that context for like really trying to do high quality science in space and like connecting the biotech industry and connecting biology to that space environment especially for the biotech industry where maybe that hasn't been as successful at like let's really get that connected. So that's where they come from. But what I really wanted to talk about and like get into and like be excited about was some of the different focuses we have, in particular the agriculture. So we've done a lot of different things with pharmaceuticals and screen, we're doing medicine, trying to develop different hardwares that do that. But I just started with Rhodium recently, and my focus in my area is in that space agriculture. And so really trying to broaden that. They already have several missions under their belt of doing different astrobotany related and plant related missions and growing plants in space. We really want to kind of expand that and really look at the possibilities of how do we really develop and do synergistic agriculture in space that both helps us to understand how we move forward for space exploration, but also how do we help do agriculture better on Earth, and how does using the space environment help improve the sustainability and the approaches that we use on Earth and the agriculture we use, and to make that a more sustainable and more viable approach as the Earth becomes a more challenged environment, and as we have all these different things that we face, and trying to do better at what we're and to be more successful in that. So some of the hardware that we have available to partner with that's already flight ready. This is stuff that's already been tested, that's already been developed, that we've used in previous missions before. This includes a variety of different kinds of chambers. And so there's various tubes, punches, or various different kinds of chambers that, depending upon the project, can help to provide access and test that. And these are we've flown seeds in some of these chambers before. We've flown microbes. There's different vials that can fit inside and different things. And so we have a different number of samples, different conditions from ambient and frozen, and different things to kind of target whatever the conditions are needed in order to provide the space environment access that's needed there, and then be able to have the ground controls that. So these are both used in the space environment, and so they've applied and they've been validated and they're provided there, but they're also valid for the same exact chambers are then duplicated in the ground controls. We're really providing them that ability to do the space side. So there's a variety of different ones with a variety of temperature ranges that they're applicable for. And you can do a common way that might happen is maybe it's frozen on the way up and frozen on the way down, but then ambient conditions during the actual mission. But there's adjustments to all that. So there's lots of opportunities and availability that would be individualized to each project as to what can be used. So lots of different options for that in order to create them together. And along with that, so something that we usually send along with any mission is the temp log, which is our current variation is the temp log 4 rad. And that this current variation can log temperature, humidity, and radiation, just like track the distributed radiation environment. And it's small and modular, so it can fit right next to the samples, and it goes where the samples are. And so it allows for that you to track the environment of your samples right there in the same area. So it's not just the temperature recordings from the other side of the room, but it's the temperature and the humidity and that environment that's right there next to the samples. And so that's really helpful in that. And that's currently rated down to -40 degrees Celsius. And so it can be frozen in not quite down to the -80 level, but it can go along in the freezer to some extent in the freezer with the samples, depending on how low that needs to go. So there's a lot of capacity there. Also, the ability to look at variable gravity with centrifuge that is aboard the ISS. So it's on the ISS ready for use, depending on the mission that's there. And it has a different variable gravity for simulated gravity. And so that can get some different options for doing that. So there's lots of options there, and that's pretty modular. It has different types of holders that you can do for that. So, and then something that I'm really excited about is one of our newer instruments is Sophia. And so I'll talk more about that's being used on a mission right now. So I'll talk a little bit about that's one of the projects I'll talk about is how we're using that. But that's a plant growth, a small plant growth chamber that provides light and
Don't do it. I
while
will powers back, you know.
and you can do up to 12, and so up to 12 individual growth chambers, so they're each isolated from each other, and you can do the different so there. And it has you can do it with plants, and you can also put microbes in it or both together. And so it has a lot of variability in what you can put on it and what can be done. And you can see here it's lit up with the current lighting that it has. And there's lots of, and this is Sophia 1, but there's future iterations that are coming out for that. So there's a lot of excited things that, and a lot of things to look forward to with that. And that's one of the hardwares that can be available for use. And the current experiment that we're using that for aboard the ISS that is Sophia Leo Integrated Floriculture Experiment or LIFE. And so this is a pretty fun one that I'm excited to see as we've been getting images from it on a daily basis, twice each day from the ISS. And so I have a couple of the images over here. On the left is just one that's just how it looks in general. But on the right is one of the ones from the ISS experiment. So some of the plants that are growing inside of it on the ISS. So you can see a little bit Arabidopsis there. And so that experiment was really looking at root growth with genetically modified plants. And so specifically a gravity insensitive gene, like an adaptation to a gene to make it gravity insensitive, so that you have these variants of Arabidopsis thaliana compared to just a wild type growing up under the ISS. And then right now we have all this imagery that we've had from that, and then we'll have the temperature and the humidity and the radiation of the log from that, as well from our temp logger, and that will give us those conditions. And then we'll just be able to use the cuvettes that the plants grow in are freezable, and so you can take the samples, you can freeze it, and that will allow us to do some transcriptomics and other genetic molecular when the samples return. So that's really cool. So I'm looking forward to that. So no data to present yet, just because it's still in action, but a really fun mission that we're looking forward to seeing the results. I know there's already a second iteration in the plan and in the works that we can talk about in a future date. That'll be in the works for that. And the second is a project that that we're getting started with. And so this project is still in that development phase, so it hasn't flown yet, but it's scheduled to fly. And so and there's a lot of really cool things happening to this one. And so this is a newer project that we're doing, really focused on the agriculture side, and challenges of how to do sustainable ag and profit, and how to connect the agricultural industry. There's a lot of research and really trying to understand how to do agriculture, both from the industry as well as the academic side, really connecting that industry to the space environment. And so trying to bring these small owned businesses from the state of, they're each from the state of Alabama. So we have several that we've selected, and putting together a collaborative mission where we actually combine a lot of different science together. We're going to be looking at everything from flying some seeds, as well as flying some different microbes to look at metabolite production, and all this combined into kind of this really fun mission planning for that. So there's looking forward. And so those are the kind of mission that applies some of the other chambers that we have, in addition to like the temperate board. And so looking at that. So that'll be, I think we're really excited to see what we're going to be able to do to help understand how the space environment can be applicable and synergistic in how we do research for agriculture in space. So I'm really excited about that. But there's a lot more opportunities and interests that we have in expanding that and moving forward, and everything from looking at regolith-based agriculture and how you develop that and finding that. That's an area that I have a lot of expertise in, and so can provide support to develop projects for those. And really trying to, and making sure that the simulants that are provided are those that are appropriate for those kind of experiments. But also, and just in general, just looking at how we answer some of these big questions that we're talking about today, and all this really cool science is taking place. How do we connect more of that science to the space environment, and get some of that commercial side that into the loop, so that we can kind of create that synergistic energy and looking at all these different things. So if any interest in collaborating or looking at developing those technologies and helping or interest in doing a project together, that's the contact information for that. And I hope that was helpful, and hope that provides a resource for all this great work that's taking place, that can access to that space.
Okay. Well, thank you, Laura. That was really great to hear from some of our industry partners. I think it's easy for scientists to get so wrapped up in the questions and the data that they forget all of the hardware and infrastructure logistics that are needed to get it there. So it's really good that we have you to show us the companies that are doing that on the daily. Oh, thanks. We got your contact information in the chat. Thanks, Chris. So while we're waiting for questions to come in, I've got one. I'm curious about your transition from academia to this kind of industry. What motivated you, and how has it changed?
It's still a pretty fresh transition, so that's a great question. But it's really, I have a passion for the science, and I really want to see the science done well. But I also know that a lot of the funding, a lot of the ways that we can do this are in industry, and so being able to connect the two together, I think, are really important, and being able to have that ability to make sure that we're doing things in a good way and that it's sustainable and we're approaching that and we're trying to do. And so I think it's been good so far. But get back to me in a few years.
Sure. Yeah, I'm curious about that one experiment is something
Yeah.
life with the Arabidopsis.
Yeah.
That device was pretty intriguing. Was it 3D printed?
Yes.
Hmm.
Yeah. A lot of it's 3D printed.
That's cool. And I guess the cuvettes are they sealed? Like, I'm just always curious about the tolerances with 3D printing and creating closed environments.
So there is a way that we do have to seal them in order to have it fly aboard the space station so that it's safe for that. So they are sealed after a fashion, yes.
Okay.
With very specific protocols that are required for space flight are put into that. And because of that, you can do like micro experiments in them as well as planet.
Okay.
It allows for that to meet the safety qualifications.
Sure. Great. Kevin's got a question, which is you mentioned regolith simulant. How well do you think that simulates regolith for plant growth?
I think that's an excellent question. So that's a really big area of mine, is that I focus a lot on regular stimulant development and specifically on stimulants for agriculture. So that was what I spent a lot of my dissertation doing, was doing that for Mars. And so I have a lot of opinions about regular stimulants, and especially in their appropriateness for use in biological applications. It's probably a long conversation that we can have right now, but most stimulants, the way that they're designed, aren't necessarily designed with biology in mind. And so that can be a very big limitation into how you apply them and to making sure that the research questions you have are appropriate for the stimulant. So that's a really important thing to consider as you're designing.
Yeah. I think I've dabbled in regolith stuff myself, and you always have to double check if it's a physically accurate replicate or a chemically accurate replicate. That definitely has huge implications for the biology in particular.
Yeah. And it really goes back to the research question that you're doing. So there's not a one size fits all answer. It really depends on the research.
Any other questions from the audience? Well, I'm not seeing any. So I guess if you guys do have any questions, follow up. Laura's email is there in the chat, laura@rhodiumscientific.com. Yeah, thanks again, Laura, for doing this last minute fill-in presentation. That was really, really clutch. I appreciate it.
Thank you.
Yeah, so here we are. The last thing on the schedule was Dr. Caswell's Humans in the Ecosystem chat, but we already did it earlier in place of Christina Escobar's Artemis Leaf Mission talk. So we're at the end of the schedule here, 20 minutes before 4. I don't see any reason to keep everyone later. I don't have anything to say. It says closing remarks there, but these are the remarks. So thanks everyone for coming out. This first day was fantastic. Really excellent stuff today. Got me really excited for tomorrow as well. So I hope to see you all back here tomorrow for another round of really cool presentations about space ecology, bioastronautics, agriculture, and everything else. Yeah, thanks. I'll see you all tomorrow.